Hey everyone, this is Dr. Jyoti Bala. I welcome you all on my YouTube channel. I would also like to thank everyone for your support, like and feedback. I really do appreciate that. Now let's begin this session. Today we will be discussing about a very important subject, which is sequencing revolution, especially the next generation sequencing. So if you are from biomedical, bioinformatics and biotech sector or from life sciences, you might be knowing about the importance of your DNA, RNAs and the relevance of sequencing. So in this session, I will be covering an overview and the scope of especially the NGS. So let's begin this session. Here I wanted to emphasize a very important point also, that we are in a time of interdisciplinary sciences. If you're coming from a biological sector or basic biology, botany, zoology, microbiology, genetic, but if you want to come to the applied or translational research or applied areas, you have to understand a lot of data related to biological sectors also. So we are dependent on bioinformatics, data scientists, IT and all these kind of things. If suppose you are working on in silico drug designing, then you are dependent on chemistry people also, you are dependent on bioinformatician, computational biologist, molecular biologist and so on. So just because you are studying basic biology doesn't mean that you don't require other subject area, and especially in this current time when we have to deal with other subjects also. So, and here a very important part is your bioinformatics and data sciences for your biologist people. So we have to make ourselves comfortable with computers, data, and now we are in a time when people from biological sectors, people from pharma sectors are utilizing very smartly computer sciences, IT, AI, machine learning, big data biology and so on for several projects. So we should enhance our skills in those subject areas also. We all know molecular biology and understanding of molecular mechanism are very important to decode and to understand any biological system, physiology and biological problem at molecular level. As we know, our genetic code lie in the DNA in most of the organism. Few exceptions are RNA viruses. So that means to understand a biological system at molecular level or to understand a molecular problem, we have to understand these DNA, RNA, the central dogmas and the RNA vault hypothesis at the molecular level. So the molecular biology plays very important role in biology, in biotechnology, and to decode these DNA and RNA at the genomics level, we are relying and we are taking advantage of bioinformatics and big data biology also. So if you see how revolutionary research and technology advancement has happened in few past decades, you can see the development and the discovery of DNA has played a wonderful and the major role in this journey. And then the cloning, genetic engineering, then the decoding and the sequencing of our human genome is very relevant, very relevant phase in this molecular biology journey. And then the CRISPR based genetic editings and CAR therapy and all these things. So in a molecular biology and bioinformatics, we are seeing a lot of advancement. But to understand these kind of work and to interpret these kind of biological data, we require to read our genetic informations, the DNA and the RNA part, whether we are coming from plant sector, microbiology sector or human, whether we have to understand the basic biology or we have to utilize these information for biomedical or applied purpose. So we have to understand the molecular biology, and here these technologies are helping us a lot. And for doing any kind of cloning, we require to understand the genetic makeup of that system in a much better way. And here our sequencing and sequencing advancement is playing a very crucial role. So data revolution is happening everywhere. and it is having a huge role and potential in life sciences sector also. Sequencing and especially omic data and this technology advancement is yielding a lot of biological data in botany, zoology, biotechnology and biomedical areas. So it is playing a huge role in biology also, but we have to understand this is not a small data, this is a part of big data biology. So we are in a time when we can understand any biological system in a much better way, and thereby we can utilize our understanding and interpretation for the applied purpose also, and to understand the system in a basic levels also. So let's discuss a little bit about the history and the overview about the sequencing technology. Sequencing is a technology by which you can read the genetic codes and genetic materials and the genetic system actually. So at the nucleic acid level, you can read these codes and thereby you can analyze and interpret the project and the subject areas of your biological system. In past, mostly we were reading these sequencing codes via maximum Gilbert, Sangers and shotgun. These technology were having few limitation in terms of efficiency, in terms of time and money and all these things. But we are in a much better time when we can perform next generation sequencing via Illumina, PacBio, Nanopore and so on. So in the upcoming videos, I will be explaining you all these things in detail. But here we should understand that next generation sequencing give us a platform, a technology by which we are in a much better scenario to read and understand our genetic makeup of any biological system in a much faster way. Price wise also, next generation sequencing is very feasible for everyone actually. So now we can understand biological system and molecular system in a much better way, and thereby utilize these information and these interpretation for various applied purposes, which we are going to discuss in the next few upcoming slides also. So if you will compare the sequencing via next generation sequencing and the Sangers or the conventional sequencing, there are few advantages when you perform next generation sequencing, like in terms of cost, reliability of data, because here multiple reads they will do. If you talk about money and the time, it is very fast, efficient and cost effective. Many people nowadays can perform NGS for diverse application. When we were doing Sanger, there you have to fragment your DNA, then you have to do the cloning, then you have to do the screening and then send the samples for the reading purpose via Sanger sequencing. But when suppose you have to perform RNA sequencing via conventional, then during that scenario you have to convert your RNA to cDNA, cDNA to again cloning, and then you have to read these things. So it is a complex and it is a time consuming and costly effort. But in case of NGS, direct RNA-seq, small fragment, large fragment reading, whole genome sequencing, parallel and multiple sequencing. So all these things is feasible, and right now we know we have to understand microbiota, metagenomics, all these kind of complex data also to understand the biological system. Everything is feasible. So it gives us a very wide platform for various applied purpose. So you can read your DNA, RNA, whole genome, small fragments, multiple sample via this technology. So obviously because of this reason we can utilize these information and data for different application purpose also. So NGS is playing a very important role, as I already mentioned. It has a huge scope and application in diverse area, because it gives a platform by which we can get the idea and the information about these genetic makeups and code, and thereby to decode the basic biology, or we can use these information for applied purpose. So whether you are working on a drug designing kind of project, personalized medicine kind of things, cloning, whether you have to utilize CRISPR based text system for editing, but prior to that we have to get the ideas about the gene, genetic makeup, mutations, what part of that genomes or genes are relevant, what need to be overexpressed, what need to be edited. All these information we get once we read these genetic informations and genomic information. After that, you can utilize these information in plant-based system. You can modify your plant, you can enhance the property of those plant, you can enhance the properties of your microorganism, you can utilize these things for your biopharmaceutical, recombinant DNA technology, for your expression of the protein, and so on. So in case of vaccine development, in case of drug designing, biomedical, plant, food technology, personalized medicine, everywhere you can utilize these NGS information. So along with your genomics and NGS data, other omic data is helping us a lot to understand any biological system in a much better way, and this is the reason we are seeing personalized medicine era, personalized agriculture, and so on. Because now we are in a time when we can understand any genetic makeup of any organism, genetic variations, epigenetics, the outer effect, the microbiota, metagenomics, all these things, and in integrated way we can understand the systems and the problem in much better way. So this is the reason NGS and other omic technology is playing a very important role in understanding the biological system and thereby using these information in applied and translational way. So NGS is helping us a lot to understand any biological system. So let's take the example of human. If you want to understand the whole genome or genomic constituent of any system, you can do the reading, whole genome reading, whole exome sequencing, and then you can check the differential things in different experiment, different sets at the molecular level. So you can understand the mutation, small mutation, copy number variation, in fact the structural variations also. So in that way, genomic studies, and then if you're working on RNA-seq or transcriptomic differential things, if you want to analyze, these things also feasible. And along with that, epigenetic differences, methylation, histolization, all these things analysis we can perform at the molecular level. So genetic level, transcriptomic level, epigenetic level, once we understand these things at molecular level, we will be in a much better condition to understand the biological processes, molecular interaction, and in the integrated system way we can analyze all these information, and thereby we can utilize these information for a better solutions, like in terms of personalized medicines, in biopharmaceutical, in designing the drugs, and also. But we have to understand the big data biology, the genomics, and this omics kind of data, what are produced by sequencing or next generation sequencing and other omics technology. They yield a lot of data, which is a part of big data biology. And we as a biologist has to understand, while doing these analysis and interpretation, you require certain tools, softwares, and pipelines. So a prior understanding with your Python, Linux, R is going to help us when you are going to deal with this kind of big data biology system, especially what is generated by next generation sequencing. So it is always advisable for biologists also to start learning these new programs and learning and languages also. Okay. So if we talk about the global market and insight, which is based on NGS, NGS is already a $1 billion industry which is booming at a very high rate, And because of this personalized medicine era, because of your biopharmaceutical, because of the relevance of genetic cloning, vaccine development, NGS is playing huge role in R&D sector also, in a biotech sector also, and in biopharmaceutical and pharmaceuticals also. So it's already a billion-dollar industry which is booming, and because of this genetic screenings, personalized medicine, cloning, CRISPR, biopharmaceutical. So in a biotech sector also, and also in the R&D sector, people are required to develop products, to do certain amount of services in terms of data analysis. So both for doing the actual wet lab kind of NGS, producing the product based on NGS, and for analysis and interpreting these data, people are required. And so technology advancement while performing the sequencing and the data which are produced by NGS, people are required both in the product development and both in the service sector also to analyze these data. So let's talk a little bit about what will be the relevance of doing and to studying these kind of NGS apart from the real applied application. If you are a young student and researcher who wanted to learn NGS and who want to come to this sector, as a research and career perspective, if you are able to understand any biological system, whether you are from plant sector, zoology sector, genetic sector or biotech sector, there will be huge scope. If we talk about the biomedical sector, you can come to diagnostic therapeutics, especially any diagnostic system you can develop, especially the genetic or the molecular based diagnostic. So very efficiently, effectively, in a smart way, we can design these things. You can utilize these genetic informations, genomic information, variant informations, RNA-seq information for designing and understanding the biology of diseases, understanding about the host, and thereby you can develop efficient therapeutics also. In clinical research also, populations wise, the effect of drug will vary. So in that terms, how the medicine is going to impact, how the pathogen is having different effect on different reason. At molecular level, we can decode all these things. Based on these things, you can develop smart bio devices also, product also. If you are good in academia or faculty, you can teach also. If you are good in NGS data analysis, you can become a data scientist in genomic sector also. And based on these things, you can utilize these informations in different biological sector, as I mentioned, in plant sector also, in food technology also. And as I have mentioned, it is going to play a very important role in biological, biotech and bioinformatical sector. And whether you want to contribute these information in basic, in applied area, depending on your research interests, you can wisely choose these things and you can come to the either therapeutic, drug designing, pharma sectors. And those people who are good with technology, who are good with IT and handling this big data biology part. So with your biology understanding and with your understanding with the biological data and data sciences, you can come to R&D sector as a bioinformatics scientist also. And you can utilize these things in developing new products also, and for new projects for your startups also. So overall, if you will see, the NGS market is huge, as we have already discussed the wide applications. In similar fashion, the scope and the market is huge, whether you want to come at the basic level or in the applied level, whether you want to develop a product or whether you as a scientific community or as an NGS scientist in wet lab or dry lab want to work in sequencing sector, or maybe you want to interpretate these data as a genomic scientist, and then you can help the plant sector, microbiology, biotech sector, in pharmaceutical, biopharmaceutical. You can either come to the product, service, data scientist, both in the academia, in R&D and in biotech sectors and in biopharmaceutical. So in a personalized medicine era, people who know these NGS experiments, who knows these data analysis will be required, and this is the reason we are seeing job revolution and sequencing revolution in this sector. So if you take the example of COVID-19 also, every month you are seeing new variants, new mutation is happening. So the understanding of these SARS-CoV-2 at molecular level is happening because of NGS and because of this sequencing revolution. Similarly, cancer-like disease, which happens because of multiple reasons, then to make a personalized medicine for that kind of disease, we have to understand its genetic makeup, we have to understand the mutations and all these things. So for genetic screening, for understanding the diseases, for developing a biopharmaceutical, people will be required both in R&D, biotech, and for in the academia also. So depending on your interest, depending on what stages you are, you can easily come to this sector. So I hope the session and the information was useful for you. If you have liked the information and this video, I kindly request you to like and subscribe this channel and kindly share these information among your scientific endeavor. If you have any query, you can drop me a message in the comment section. I would be more than happy to reply you back. Thank you so much.